Hey there, SF. My name is Guy Grewal, and I'm from Springfield, Missouri here in the United States. My project dealt with analyzing exactly how PFAS chemicals interact with cellular organisms. Because in the 2023 report, 97% of Americans were found to have toxic levels of these PFAS chemicals within their blood. But the question is, how exactly are they affecting our body above just cancer and liver disease and thyroid disease? So my project dealt with analyzing how a single type of PFAS, PFOA, interacts with yeast cells, Saccharomyces cerevisiae, because these yeast cells share 20 to 30% of their genome with humans, making them an effective model organism to study. I conducted an RNA sequencing at a 1.6 millimole concentration of PFOA and at a non-treated control concentration. I took 6 samples of each and sent those to a biopharmaceutical lab specializing in RNA sequencing. They came back to me with a huge block of data. This block of data contained hundreds of thousands of different genomic sequences outlined by Saccharomyces cerevisiae that are harmed by PFOA. So then I concatenated all that data, trimmed all the redundant sequences and genes, and then isolated to the most significant genes affected at a p-value of less than 0.05 and a log change greater than 1.25 and less than negative 1.25. So then I plug those into KEGG pathway databases, allowing to see how they affect biological processes. When it comes to the ATP production, we're seeing oxalosuccinate is dramatically affected. That's key for producing ATP, and when that's affected, our body can't produce it. So that's one way of harm we're seeing. But then we take a step deeper at purine metabolism. When it comes to histidine and diamine metabolism, that's key to DNA repair and reconstruction. And we're seeing significant harm there. But take one step deeper, G1 and S phase part of the cell cycle, we're also seeing significant harm. So it goes to show that PFOA is really causing harm in stress response pathways as well as metabolic pathways, including DNA and RNA repair.